are kind of important for you to know as we go forward into some of the more difficult stuff. So genetics started with Gregor Mendel in the 1800s. You probably know about this, where he crossed a big tall plant with a real short plant. And then he got offspring that were all tall. But then when he took two of those daughter generation plants and crossed them, all the offspring were tall except for one. 25% of them came out short. And so he realized that there are what's called recessive alleles and dominant alleles, and they pass through the generations in the population. But let's talk about what that means for a second. I'm going to probably go over a little into meiosis tomorrow, but we have 23 pairs of chromosomes, 46 total, but 23 pairs, 23 from mom and 23 from dad. But of the pairs, they're the exact same type of chromosome. It's just you got one from your dad and one from your mom. And on the chromosomes are where the genes are located. Remember that chromosomes are just wrapped up DNA. So the segment of DNA that might correspond to a gene is the exact same that you get from your dad and mom in terms of the location. Now, you might get a different form of the gene, though, from mom or dad, and that's what we call alleles. So you've probably seen there might be AA or little a, or big A, big A. If you have these, where you got the same exact form of the gene from each mom and dad, you're called homozygous. But if you have one different from each mom and dad, you're called heterozygous or hybrid, or sometimes referred to as a carrier, because you got the different forms of that gene. However, one of those forms is dominant, might be from mom or dad, whichever. But one of them is dominant, and the gene is usually expressed with that dominant trait. Now, this is the very simplest form of genetics. There are things that get much more complicating, even beyond AP Bio. And if you take genetics in college, which if you're a biology major, you'll get into some really deep stuff with that. But again, you get one form of each chromosome or gene, if you take it down to that level, from each of your parents during meiosis. And there's all kinds of different combinations that you can get from mom and dad. That's why, and that happens during the formation of sperm and egg cells, and that's why you and your siblings might be different. You're all different combinations of different genes from all different parents, from both different parents. And so you get these two different alleles, and now we're gonna talk about Punnett squares a little bit. You've probably made these. These should be all reviewed. Nobody should not know how to do a Punnett square by this point in taking AP Bio. But if you do, what we do is we put the mom on one side and the dad on the other, and this allows us to explore the combinations of possibilities for traits that offspring may have. So if we have, let's say, rabbits, and black fur is dominant to white fur. So if you have big B, you're black. If you have big B, you're black, but you're a carrier, but you're still black. But if you have little b, you have white fur as a rabbit. If we cross two black rabbits, but they're both carriers, or they're both heterozygotes, we can fill in the Punnett square by taking one from mom, one from dad, and looking at all four possibilities of genes that you could have, of allele combinations that you could have. And this gives us probabilities. And so 3 of the 4 boxes, you have a 75% chance of being black, but 2 black-furred rabbits can give a 25% chance of being white-furred, as long as they were heterozygous to begin with. If one of the parents was homozygous big B, then all of them would have been black. But this just gives us probabilities. It allows us to see the combinations. Now, I do want to give you 2 terms that might be on the AP test. The letter combinations are called genotype. The actual trait is called a phenotype. So genotypes are the letter B, but the actual phenotype is black fur versus white fur. If we did a black heterozygous parent with a white rabbit, we would fill it out like this, and you would get a 50-50 chance of being black, 50% black, 50% chance white fur, okay? So that should be reviewed. Everyone should know how to do those. There are a couple things that make those a little different sometimes, so I want to talk about the exceptions a little bit. One of those is called incomplete dominance, and that's when the dominant trait, the hybrid, is going to be a mixture of the dominant and the recessive trait. So, for example, if we have roses follow this, red and white roses, normally the heterozygote you would think would be red as dominant. So we have RR, that's going to be red. The little r is going to be white. But this one normally would always be red, but in this case, roses are incomplete dominance with the terms of their color. So these are actually pink, which is a mixture. And so that makes it different. Same concept, you can make these just as easily, but anytime you have the heterozygote, it's going to be the mixture of the two, and that's incomplete dominance. Maybe another example would be tall and short, but then the hybrid is medium-sized. Sometimes that's an example of incomplete dominance. But if we took like two, I don't know, let's say if we mix a red flower with a white flower, you're going to get 100% of the offspring actually all be pink. But if you did, let's say, two pink flowers, which is like this, you're going to get 25% of the offspring red, 50% of them be pink, and 25% of them be white. So that's incomplete dominance. Another example is called codominance, which is sort of very similar, but the hybrid in this case shows whatever the dominant and the recessive is at the exact same time. So maybe you would have blue fish and gray fish, but the hybrid is going to have blue and gray on it. Instead. Or you might have like a cactus. I think an example would be a cactus that follows this, where there's spiky thorns on the cactus as dominant, round thorns on the cactus as recessive, but when you have the hybrid, it shows both spiky and round thorns on it at the same time. And so that's codominance. So those are different examples of some exceptions to Punnett squares. But again, filling out the Punnett square should be easy. You put both alleles up there from one parent at the top, one parent on the side. Look at your combinations, and you can do those out of a percentage, 25, 50, 75% chance that the offspring is going to be something in particular or not. The final one, I'm not going to actually go through those codominants. But the final one is sometimes there are more than two alleles. So far it's been all two alleles. There sometimes might be examples where there's more than two that are contributing to the phenotype of that organism. For example, blood types is the best example. You can be blood type A, B, or O. If you're A, you have A, IA, or you have IA and just a little I. Either one of those is both for A, okay? You could have B where it's IB, oops. Like that. AB is gonna be IA with an IB, and O is just gonna be two little I's, okay? So A and B can each carry that one little O allele, or they could be completely dominant. But see how there are multiple different alleles here? There's more than just two. There's actually three, and so it just matters which ones you get. But you can do Punnett squares to figure this out as well. So if we were to like make someone have blood type A, where they have the IA and the O, they're still blood type A, and we mate them with an O-type blood person, you're going to have 50% of them be blood type A, and you're going to have 50% of them be blood type O. Or you could do, let's say we do a blood type A person with a blood type B person, but they both carry the O allele as well. Then 25% of them are going to be blood type AB. 25% of them are going to be blood type B, 25% of them are going to be blood type A, and 25% of them are actually going to be blood type O. So even though you have an A parent and a B parent, some of the offspring could be blood type O. Now, if you make two O people, all the offspring are going to be O no matter what. They have to be. If you have someone who's dominant, then all the offspring are going to end up being like the dominant. But that's just another example, and those are where you have multiple alleles. But anyway, that's all I kind of wanted to go over today. I'm going to post a practice problem set right now on Google Classroom for you to do over the next 24 hours to practice, and then at some point I'll give you the answers. For this one, I'm not going to have you turn it in. So many of these times I will have you send it to me and turn it in, but for this one, you do not need to turn it in. But I would like you to write them down and save